Hello, very good morning to all of you. I hope you all are happy, healthy, and fine at home. So, what did we discuss yesterday? Yesterday, we discussed about the peptide synthesis, how peptides are selectively synthesized, and how we are getting the desired product that of the thing we have seen. And today, what we are going to discuss? Today we are going to discuss about protection and deprotection. We are learning or discussing. Among that there is a nucleic acids, and as we know that, what do you mean by the nucleic acid? Who first time invented this nucleic acid or first isolated from cell nucleus, hence name is a nucleic acid is given to that species. These are polymer molecules, and there are two types of, as we know that there are two types of nucleic acid. Number one, first one, what is that? First one is known as RNA, and this RNA is found in cytoplasm and nucleus. This is found in cytoplasm and nucleus. That is a first type of nucleic acid, which is RNA, and second type of nucleic acid, you already familiar with this type, that is a DNA, right? DNA, and that DNA found only in nucleus, found only in nucleus. So in a nucleic acid there are two types. One is RNA, which is present in cytoplasm. Let me write that word properly over here. Cytoplasm. So which is present, found in cytoplasm. This RNA found in cytoplasm or found in cytoplasm. So as we know that nucleic acid, and this nucleic acid, let us see here, nucleic acid, after the hydrolysis, what do you get? After the hydrolysis, what you are going to get? You are going to get nucleotides. You are going to get nucleotides. And as we know that nucleotides further hydrolyze is one hydrolysis of these nucleotides. What do we get? We get nucleosides. We get nucleosides as well as phosphoric acid, H3PO4, and this hydrolysis in organic acid. This one further hydrolysis in organic acid. The resulting form we are getting here is a base as well as sugar. So this is a general things are happening here. Look at, we just now seen and nucleic acid further divided into two types. One is called as RNA, which is found in cytoplasm and the nucleus, and second type is known as RNA, and this RNA found only in nucleus. RNA means ribonucleic acid. DNA stand for ribonucleic acid. So that are the full form of RNA and the DNA. And this nucleic acid, this is for the nucleic acid when hydrolyzed, it is converted into nucleotides, and that nucleotides further hydrolyzed, we obtain here nucleosides and the phosphoric acid, S3PO4, phosphoric acid, and that nucleosides further hydrolyze in organic acids. We are getting two components over here. One is a base and second one is a sugar, and hence the base and the sugar are the starting material for the formation of nucleic acid. Remember here, base and the sugar are the starting material used for the formation of nucleic acid. Now let us see here what are the bases and what are the sugars present over here. Now let us see here in that base. Just now we have seen the starting material used for the formation of nucleic acid will be the base and the sugar, right? Now this nucleic acid we have just seen there are two types. One is RNA and second one is a DNA, right? Now the starting material used for the synthesis of nucleic acid will be the bases. Look at here the bases, and that bases further divided into two types. Number one, here I am writing, number one is known as purine bases. There is a purine bases, and the second one is known as a pyrimidine bases. Second one is known as a pyrimidine bases here. Right? Base further divided into two types, or there are the two types. One is called as a purine bases, second one is called as a pyrimidine bases. As we know that bases means what? The organic compound containing nitrogen atom are basic in nature. This is a general guideline, or this is a general statement regarding the organic bases. And as we know that most of the compound containing nitrogen atom, and why they are basic in nature? As we know that lone pair has the ability to donate the lone pair to the other species. Normally the H plus are known as a bases according to as a Lewis theory. So this is a purine bases, and these are the pyrimidine bases. Now let us see what will be the structure of purine. So structure of purine is this one. Let us see here, see carefully the structure of purine. This 5-membered ring joined to the 6-membered ring over here. This is a structure of purine. This is a structure of purine. Look at this structure carefully. This is a structure of purine, means 6-membered ring fused with a 5-membered ring. In both the ring, two nitrogen atoms are contained in six membered ring. There is a 2 nitrogen atom it has, and 5 membered ring also. They are the nitrogen atom, remember always they are present at 1,3 position. So this is the structure of purine, right? And now pyrimidine. Now pyrimidine base means what? The pyrimidine is nothing but is a only 6 membered ring, and the nitrogen atoms are present at 1,3 position. So look at the structure of pyrimidine. Again is a very simple. Here N, then at 1,3 position you just what we can call it as a put the nitrogen atom. That is this is just like as a benzene ring. As we know that benzene is an aromatic compound. These are the heterocyclic aromatic compound. This purine also aromatic in nature. This is also as aromatic nature because they fulfilling the criteria of aromatic. They are cyclic in nature, planar, and fully conjugated. Alternative carbon-carbon single bond and the double bond, and therefore they follows the Huckel's rule 4n plus 2 pi electron, and therefore they are the aromatic in nature. Look at this one, purine, and this is a pyrimidine. Observe these two structure carefully. Now this again purine further divided into 2 types. This purine further divided into 2 types. One is known as adenine. This is known as a adenine, and another one is known as guanine, and second one is known as guanine. One is adenine, second one is a guanine. And what will be the structure of adenine? What will be the structure of guanine? Let us see here. This is a structure of purine. This remains as the basic skeleton as it is, only some functional groups are changes. The basic skeleton remains as it is in the adenine only. Now before that, let me tell you the nomenclature, numbering system, how the numbering system is given to this type of the compound. Heteroatom always getting as a number 1. Therefore, look at here the number. This nitrogen atom should get the first number, then second one, this one is the third one, fourth one, fifth one, 6, 7, 8 and 9. So this is the numbering system is given to this purine, and for this the numbering system may be start from this or this, it doesn't matter, but let us start from here. This is a number 1, that is the second one, third one, fourth, fifth and 6th. That's it. So these are the numbering system you should remember while talking about. Now the adenine structure. What will be the structure of adenine? This skeleton, this basic unit remains as it is in the adenine. This one, let me draw the structure here clearly. So let us see the structure of adenine is this one, N, then at 1,3 position there will be here N, then N, this H over here. Now look at here, everything remains as the same. Here double bond, only here in adenine there will be the amino group at 6th position, and this is the structure of adenine. What is that? Only the amino group is joined at 6th position, so that is the structure of adenine. And now look at the guanine. guanine again, the structure of guanine, the skeleton remains same, here N, then here N, this, and 5-membered ring, that N, this one is a H. Now what is happening here, there is a carbonyl group inserted over here, and here is a amino group NH2, and other things remains as it is, double bond over here and this double bond over here. This is a structure of guanine. This is a structure of guanine over here. Look at, only the basic skeleton remains as it is. Carbonyl group at 6 position, and at 2 position there is a NH2 group, that is the changes. So this is about the purine bases. Purine has this structure, and it is further divided into 2 types, adenine and the guanine over here. Now moving toward as this one, pyrimidine, and that pyrimidine further divided into 3 types. This pyrimidine is divided into 3 types. Number 1, number 2 here we can write, and number 3 you can write here. And number 1 is called cytosine. There is a cytosine or cytosine. Second one, thymine. Second one is a thymine. And third one will be the uracil. So these are the 3 bases of pyrimidines. Has this skeleton remains as it is, 6-membered ring, and nitrogen atom are 1-3 position that you have to remember, and they are here the structure of cystosine will be, the skeleton remains as it is, this one N, the nitrogen atom at 1-3 position, but in this case at 4 position there is a NH2, and at 2 position there will be the carbonyl group, and the remaining one here double bond, here is a double bond and 1 hydrogen atom satisfy the valency of that nitrogen. So this is a structure of cystosine. Here 1-3 position there are 2 functional group, one is a amino group and one is a carbonyl group. They are inserted or present at 4 and 2 position. Now the structure of thymine. Let us draw the skeleton of this. At 1 and 3 position there will be the nitrogen atom, and at 4 position there will be a carbonyl group. At 2 position there will be again as a carbonyl group, and here is a CH3 double bond, here alpha beta unsaturated compound, and here is a one of the hydrogen atom, and here again one of the hydrogen atom. So this is the structure of thymine. Look at 4 and 2 position, carbonyl group is inserted, and here is a methyl group, that is a alpha beta. Here you can, this is a just like as a this urea is inserted alpha beta unsaturated compound. So uracil has the same structure over here. Uracil again, let us draw the skeleton of this pyrimidine as it is. Here nitrogen atom, then here nitrogen atom. Now there inserted carbonyl group, alpha beta unsaturated, here is a carbonyl group, and other thing remains as it is. Here is a hydrogen atom and here is a hydrogen atom. look at, these are the pyrimidine bases, cysteine, thymine and the uracil. Here purine has a 2 type, adenine, guanine, and this pyrimidine has a 3 bases, cytosine, thymine and the uracil. Look at their structure. Thymine and the uracil has a same structure. At 2 and 4 position there will be a carbonyl group, but here look at, here there is no methyl group, but in thymine there is a methyl group. This is a trick you can remember for remembering the pyrimidine structures. And as we know that DNA and the RNA, in DNA this adenine, guanine and cysteine, they remains common. Let us look at, suppose here is a DNA is there, DNA and RNA. As we know that these are the two types of nucleic acid. In DNA what are the bases present, and whereas in RNA what are the bases present, the common thing remains adenine, guanine and the cysteine as it is. Adenine, guanine and the cysteine means AGC. Here also in case of the RNA, A, G and C remains common. Only what is changes there? In a first case, in a DNA, thymine is present, AGCT, whereas in RNA, AGCU is a present, that is known as a AG, means these are the bases combined together and we are getting as a this type of the base. Understood this one? Now let us move on to the next one, that are the sugars. Just now we have seen these are the bases. Now let us see the sugars over here, because bases and the sugars are the starting material for nucleic acid. Just now we have seen there are the two types of the bases are present. One is a purine and another one is a pyrimidine. Purine again further divided into two types, adenine and guanine. Similarly, pyrimidines further divided into three types. One is a cysteine, second one is a thymine, and third one is a uracil. And now let us look at as a sugars over here. Sugars. And now in the sugars, sugar also two types. There are two types of the sugars are present. One, here let us see here. First one is known as ribose. First one is known as a ribose, and second one is known as second prime deoxyribose. They are the 5-membered ring. Now what will be the structure of ribose and 2' deoxyribose? Deoxy means there is a the removal of oxygen, or there is a deficiency of oxygen. That's why it is termed as deoxyribose. Let us see as a ribose means this is a 5 carbon atom present in that 5-membered ring. Let us see as a furanose type of the ring is there here. Let us see. Let me draw the structure properly over here. Let us see this one. And here this HO, then here you can write OH, and here also as a OH, and here OH. This 1, 2, 3, 4 and 5 carbon atom, 5 oxygen atom, 1, 2, 3, 4 and 5 carbon atom, and they are designated as, this is known as 1', 2', 3', 4', and 5'. That are the designation of this sugar unit ribose. This is known as a structure of ribose. Now is a 2 prime deoxyribose is a structure, is a skeleton is the same over here. Here is a OH, and here OH, this is a HO. Then look at here, 1 prime, 2nd prime, 3rd prime, and 4th prime, and 5th prime. Look at second position, there is a no hydroxyl group, and therefore it is known as a deoxyribose nucleic acid. Do we know the DNA, right? DNA means what? This one, and RNA means what? This one, ribose nucleic acid, and D means what? Deoxyribonucleic acids. So that are the two main sugars present over here. The sequence of base, is nothing but as a gene. Sequence of base is nothing but as a gene. As we know that genetic information, all information we are getting from the RNA. This is a sequence of base gene, and 3 bases code for protein synthesis, and 3 code for protein synthesis. And now let us look at the next one is a nucleosides. What do you mean by the nucleosides? We are talking this just now we have seen as a what we can call it as a sugar as well as bases. There are the 3 types, how they are numbering systems are followed there over here. And I think with this let us stop today here. Remaining nucleotides that we will see in the next lecture tomorrow. Have a nice day. Thank you.